Thank you very much for this kind introduction, Kristian, and good morning, good afternoon, or good evening, everyone. To be honest, I'm very proud to be the first speaker of this talk series, and I congratulate you also, Kristian, because I think that this LiveNano platform is a very nice initiative to generate new interdisciplinary synergies that are essential for liquid phase or more generally in situ analysis. So to stick to this idea, today the goal of my talk will be to illustrate the interdisciplinary potential of liquid phase TEM by showing you some application of this technique in materials, life, and earth sciences. So as Kristian told you, I work in the MPQ lab, which is located in the southeast district of Paris. And over there, my group is working on the fabrication of nanomaterials, but we are also running a national user facility for electron microscopy, including this double-corrected TEM. So the goal of my research is to exploit the multifunctionalities of this great instrument to establish the link between the atomic structure of materials and their properties. And of course, although TEM is a fantastic tool to reveal the secrets of matter at the atomic scale, studying materials in vacuum is not enough to fully understand their behavior. And nowadays, we know that there is a crucial need to image and follow the dynamics of materials in realistic environments. And that's the reason why about 10 years ago, we start working with liquid and gas cell TEM. So for those which are not familiar to these techniques, the idea is to exploit the multifunctionalities of transmission electron microscopy to study the dynamics of materials in a liquid or gaseous environment that is encapsulated between two silicon nitride heat chip, silicon nitride wafer on which you have a window that is etched, and you can, on which there is a silicon nitride film that is transparent to the electrons, and you can control the temperature of this environment, also the composition, by injecting any kind of gas or liquid at the observation window through a microfluidic system that goes through the holders. So today I will be only talking about liquid cell TEM, so all the experiments that we do in liquids, and I'll show you what we can do with this technique to study the nucleation and growth of nanomaterials, to study the structure and dynamics of biomaterials, and also how we can study biomineralization process in realistic media. So I start with the nucleation and growth of metallic nanoparticles. Over the last decades, there has been a lot of work to study the growth with liquid cell TEM, the growth of complex nanostructures, including gold nanoplates, dendritic nanoparticles, nanostars, nanocubes, nanowire, core shell nanostructures. And all those great studies provide fantastic information on the role of seeds that are incorporated in the media to control the growth, on the role of defects, or on the growth, the role of functionalization on the growth process. But so far, the role of temperature has been disregarded. Although we know that temperature affects all the atomic scale process that drives the size, dispersion, and the shape of nanoparticles. And the reason for this is because the technology is a temperature-controlled liquid cell that allows controlling the temperature in the liquid cell. It's a very recent developments. So here I'm going to show you what we have done using the prototypes temperature-controlled liquid cells, in which you have a resistance that is embedded inside the silicon wafer that allows to homogeneously heat up the whole liquid inside the liquid cell from room temperature to 100 degrees. Now keep in mind, all the process I'm going to show you here, they are driven by radiolysis. So I'm going to show you experiment where we expose to the electron beam an aqueous solution of chloruric acid, and due to the radiolysis of water generated by the beam interactions, you have this radiolysis process generate a lot of oxidizing and reducing species, And the most reducing species are the aqueous electrons and the hydrogen radicals that reduce the metallic precursors to the atomic states, to the metallic state, and generates the nucleation and the growth of nanomaterials. And what is very interesting with this technique is that we can finally control the dose rate, which allows us to control the growth kinetics, because the dose rate is directly related to the concentration of reducing precursors. So that's something you always have to keep in mind when you perform liquid cell TEM. You have an effect of the beam, and I illustrate this with these two examples here. One growth experiment of gold nanoparticles performed in low dose STEM imaging, and one in high dose TEM imaging. It also illustrates how we can study the growth process at different scale, ranging from the whole colloidal assembly to single nanoparticles up to the atomic level. But you see that the growth is drastically affected by the electron beam, and you have completely different growth process when you use low dose and high dose. So of course, if you want to study the effect of temperature, it's very important to compare experiment performed with the same electron dose rate. So that's what I show you here. Here you see 3 series of experiments performed at 25 in low dose condition at 25 degree, 50 degree, and 85 degree. And when we saw this result with Abdelali Kelfa, who is the PhD student who collect and analyze those data, we were very impressed by the effect, the drastic effect of temperature in this low temperature range. You see that at 25 degree, we grow a very dense assembly of small nanoparticles. At 50 degree, we reach roughly the same size of nanoparticles after only 30 seconds, whereas here you see it's 75 seconds. And at 85 degree, we have only a few tenths nanoparticles that grow, a few tenths of very large nanoparticles that grow in the liquid cells. So of course, if you want to be quantitative, you need to use automated video data processing. That's what we do. We did it using Fiji, and also this very nice platform developed at EFPEN in France, Plugin. It's a freeware, so it works very well for our systems. And this allows us to quantify the number of particles over time inside the liquid cell during the different experiment. And we clearly quantify the drop in nanoparticle density with temperature. You see that after the end of the nucleation phase at 25 degree, we have about 900 nanoparticles. At 50 degree, about 300, and only about 10 nanoparticles over the same 45 micron square area that we observe at this magnification. So this result is actually but something that is also interesting here, if you compare the nucleation rate for the experiment performed at 25 and 50 degrees, you see that they are roughly the same. So it tells us that in fact the decrease in the nanoparticles density is due to the shortening of the nucleation period when you increase the temperature. So now if you look at the nucleation theory, you realize that this result is not so clear, because if you look at the nucleation rate of nanoparticles as a function of the supersaturation for different temperature here, you see that we expect to have a higher nucleation rate at high temperature, which is not in good agreement with what we see. So the fact that we have a shortened nucleation period at high temperature means that the supersaturation ratio here decreases faster at 85 degrees than at 50 or 25 degrees. So now we have to think about what is this supersaturation ratio. So this is the ratio between the concentration of gold precursor in the observation area and divided by the equilibrium solubility of the solutions. And something interesting here, by quantifying the total volume of nanoparticles, we have an idea of the dynamics of the concentration of gold atoms, because of course these are all the atoms that has been consumed, so they are not anymore available for the nucleation. And you see that on this graphic, you see the evolution of the total volume of nanoparticles as a function of time, and you see that the concentration of gold atom in the liquid cell will decrease faster at high temperature, which could explain why the supersaturation ratio decrease faster at 85 degrees. But still, if you look at now the quantity of materials that has been that has been formed, the quantity of gold that is inside the nanoparticles at the end of the nucleation period, you see at roughly 30 seconds in the at 25 degree, about 9 seconds at 50 degree, and after 1 second at 85 degree, you see that there is still much more materials that has been consumed at 25 degree than at higher temperature. So this means that this decrease, this dynamics of the concentration of gold atom is not enough to explain our results. And there is probably also an effect on the equilibrium solubility. And actually, the equilibrium solubility of solution is known to increase with the temperature. And you can check that at home by doing a very simple experiment where you put salt you put salt into water and you reach the saturation level. So here you have a saturated NaCl solution. When it's cold, you have a lot of salt at the bottom of your glasses. And when you heat up this water, you'll see that we can dissolve much more salt inside your water. So we know that this equilibrium solubility increase with temperature, and now the idea is how we can quantify that to explain our nucleation dynamics. So to do that, we have observed the growth dynamics of the nanoparticles. So here you observe the logarithmic relationship between the mean radius of all the nanoparticles over over time, as a function of time, and you see that the growth of nanoparticles at these different temperatures, they follow in fact the same growth trajectory, where we have a linear relationship between the time and the cube of the mean radius. And this is very consistent with, so it means that, of course, the growth dynamics increase is much faster at high temperature, but it does not affect the power law exponent, you see, which is pretty much the same. It's only affects the growth rate constant K here in this equation. So these results are perfectly in line with the diffusion limited growth model that has been developed a long time ago in the LSW theory. And if you go through this theory, you realize that you can have an expression of this K factor, the growth rate factor, that depends only on the solubility of your solution and the viscosity of water. Which is known in this range of temperature. So from the variation of K that we have measured on this experiment, we have been able to deduce the change in solubility as a function of the temperature. And with this new information, we can now revisit the nucleation theory, taking into account the variability of the equilibrium solubility, which affect, of course, the supersaturation ratio. And now you see that the two curves of the nucleation rate as a function of the supersaturation ratio overlaps at 50 and 25 degree, which is perfectly in line with the fact that we observe the same nucleation rate during the nucleation phase at these two temperatures. And you see now that the decrease, the nucleation rate drops much more rapidly when the supersaturation decreases at 85 degree, which perfectly explains why the nucleation period is so short at 85 degree. So to the end, you see that the liquid cell TEM allows us to quantify the effect of solubility on the nucleation and growth dynamics of nanoparticles, which is obviously very important to understand what's going on in colloidal assemblies. Now, one of the main interests of liquid cell TEM is that you can directly see what's going on, so you can go beyond the well-established theory of nucleation and growth. So as the behavior of the nanoparticles is very consistent with the LSW theory, we know that Ostwald-Rippning is at play in the process, and we have obviously metallic transfer from particle to another. And what is interesting here is that it's only at 85 degree. Here I show you an experiment performed at 85 degree. It's only at this very high temperature that we can clearly see what we call Ostwald-Rippning, meaning the dissolution of some particles that shrink at the expense of the other ones that grow. And the fact that we only observe that at 85 degree is also an effect of this solubility, meaning that the nanoparticle solubility increase with temperature. So if you look at the textbook, you will probably read this idealistic view of Oswald-Ripning, which says that the growth of the larger particles, the Oswald-Ripning is the growth of the larger particles at the expense of the small one. And in this experiment, we realized that this common view of Oswald-Ripning is not completely true, because on average, yes, the particles that shrink have a mean size that is smaller than the particles that grow. But when you look at now, it's well exemplified in this example, you see that you have also a lot of large particles with a size way above this mean radius 55 nanometers that can shrink too, which clearly show that the Oswald-Ripning is not only driven by size effect, and we can reasonably assume that probably the presence of defect in some particles will clearly affect their degradability. So it's not only size that rules the dynamics of nanoparticles. Dynamics of nanoparticles when you talk about Oswald-Ripning. Now, the last thing I want to show you with this experiment is how we can study the effect of temperature on the faceting process of nanoparticles. And there has been the Professor Xia from Georgia Tech University some years ago has introduced a very interesting concept to understand the faceting process of nanoparticles. It clearly demonstrates that the ratio between the shape of nanoparticles is ruled by the ratio between the deposition rates on the surface and the diffusion rates of atoms on the surface. And this figure shows you that when you have a deposition rate that is much smaller than the diffusion rate, then you give enough time to the nanoparticles to reach equilibrium shape and you stabilize a lot of 1-1-1 phases. At the opposite, if the deposition rate is much higher than the diffusion rate on the surface of nanoparticles, then you start having branched nanoparticles with a lot of high index facets. And in the middle, you have an intermediate regime where when the deposition rate and the diffusion rate are roughly the same, you stabilize cubic nanoparticles when you start from cubic nanoseeds. Now, what is very interesting in our experiment is that we can actually control this ratio between the deposition rate and the diffusion rate at the surface of the nanoparticles, because the deposition rates increase both with the dose rate and the temperature. That's something we have seen in the previous experiment. But the diffusion rates increase only with the temperature. So by playing both with the dose rate and with the temperature, we can actually modulate and tune this ratio, and we can clearly see the effect on the shape of the nanoparticles. So that's what you see here. If you are in low dose condition, meaning that your deposition rate is very low, you give enough time to the system to reconstruct thermodynamically stable surfaces, and either at low temperature or high temperature, you form very well faceted nanoparticles. And we did a lot of ex situ analysis on these particles. They are mainly made of 1-1-1 surfaces. If you work now at high dose, at low temperature, you'll start, oh, sorry, this one. You'll start forming dendritic nanostructures with very rough surfaces, which probably expose a lot of high index facets. And the reason for that is because now, because the deposition rate is very high, with respect to the diffusion at the surface of the nanoparticles, you don't give enough time to the system to reconstruct stable surfaces. And we can also observe the intermediate regime that is here, where in which you are at high temperature with high dose rate, and in this case you see the formation of a lot of nanorods and a lot of nanocubes, with, of course, which are made of 1-0-0 facets. So you see that with this kind of observation, we have now a great tool to observe the nanophase diagram of nanoparticles during their growth, which is obviously very interesting to better understand the dynamics of colloidal chemistry, of colloidal assemblies. The second study I want to show you on that topics is it's something that electron microscopists have to do to maximize the impact of liquid cell TEM in material science. We really need to catch the attention of colloidal chemists. And to convince them, we really need to reduce the gap between nanoscale in situ observation and bench scale synthesis. And a good way to do that is really to prove that the process that are observed in the liquid cell, they are exactly the same as what's going on in their beaker. So to work on this problem, I collaborate with Cyril Hamon and Deroux Constantin from the LPS at the University Paris-Saclay. And we work on a very, very simple synthesis process in which you start from a gold seed, which are bipyramidal nanoparticles, and you add only at 60 degree a silver precursor, ascorbic acid, which is a reducing agent, and you have the functionalization, which is the CTAC. And working with this, if you wait for 1 hour, you see that you transform your bipyramidal seed into very nice and long nanorods. So my colleagues, they are experts in real-time SACs and real-time absorption spectroscopy analysis, and they follow at the large scale, at the bulk scale, these synthesis, and the SACs results can be interpreted by the growth of cylindrical nanoparticles together with a lot of small spherical nanoparticles. And the cylindrical nanoparticles, they have a constant width. And now if you look at the absorption spectroscopy result, when you you see that first you have here, you look at the longitudinal plasmon as a function of time, and you see that first you have a blue shift, and then we observe the red shift. Now, with this, you can, of course, know a lot of things about your synthesis, but the idea is to compare what we observe with the same protocol in the liquid cell. And we were very happy to see that we had our nanoscale observation perfectly match what we observe with using global analysis techniques. Since we observe here, that's what you see on this video, we observe the longitudinal anisotropic growth of silver on the nanorod together with the formation of a lot of very small silver nanoparticles. And we have been able to identify that the growth is in fact a two-step process. First, you have the deposition of silver from the common base of the B pyramid toward its apex. And this actually transforms the 100 edge that you have on the common base of the B pyramid, transform this edge into facets, and that's the reason you transform, in fact, the B pyramids into a nanorod which conserves the 5-fold symmetry. And during this time, you see that the length of the at the early stage of the growth process, the length of the nanorod does not increase. And once you have transformed the B pyramid into a nanorod, you start observing the growth and the elongation of this nanorod. And by observing and determining this two-step growth process, we have been able to understand why now we have a blue shift. And my colleagues have performed some beam simulation starting from nanorods. When you transform starting from bipyramidal seeds, when you put silver and you transform this bipyramidal seed into a cylinder, into a nanorod, you see that this correspond to a change, reduction of the aspect ratio, which lead to the blue shift. And then when the nanorod is increased in size, you have the red shift. So with this, we see that we can visualize all the intermediate structure and better understand the reaction dynamics of these nanorods. Now, something very interesting with the multi-scale investigation approach that we have developed is that we can really have a new vision of the kinetics and thermodynamics effects on the growth process. So you see that when you observe the synthesis in the TEM, because the growth is also driven by radiolysis, the growth process is very fast. You have some variability, but whatever the growth speed of those nanoparticles, they always we always obtain those very nice nanorods. And what is very interesting is that in the beakers, my colleagues, it takes about 1 hour to transform the bipyramidal seed into the nanorods. So we clearly see that the reaction kinetics does not affect at all the final shapes of the nanoparticles. So it tells us that the anisotropic growth is not governed by kinetics, it's in fact governed by thermodynamics. Now we can ask the question, in this very simple chemical protocol, which chemical is the stabilizing agent? And you can think at the first, your first guess, I guess, will be say that the CTAC as a functionalizing agent is the stabilizing agent, and it controls the anisotropic growth. But in fact, we demonstrate in this experiment that this is not true. In fact, this is the ascorbic acid that drives the anisotropic growth, because we perform the same growth experiment this time without ascorbic acid. Here, the growth is only driven by radiolysis in the liquid cell. And we see here that now the growth is not it is not anisotropic, it's purely isotropic, which tells us that finally the ascorbic acid is not only a source of electron, it's not only a reducing agent, but it's also a shape directing agent. So we have identified a new role for these chemicals, a widely used chemical in liquid cell, synthesis protocols. Now, of course, the idea of our study was really to correlate nanoscale information, nanoscale study with large-scale synthesis. So we had to confirm this role of ascorbic acid with bulk-scale synthesis performed by my colleagues. And they did that by replacing the ascorbic acid by another mild reducing agent, which is hydroquinone. And they have also replaced the CTAC by sodium polystyrene sulfonate. And because they were worried about the role of this counterion in the CTAC, they also add in some synthesis some KCl to have the counterion, the Cl, the chlorus ions. And here you have the result of all those syntheses, and you see that it's only when we have ascorbic acid with the counterions, with the chlorus ions, that we obtain the anisotropic growth of nanorods. So this clearly confirms our observation at the nanoscales, and we can say that the ascorbic acid plus the counterions of the CTAC play the role of shape directing agent in this synthesis. So it's also a nice illustration of what the type of information that you can obtain by directly observing the growth at the nanoscale. So now I completely switch of subject, and I start talking about the application of liquid cell TEM to study the structure and the dynamics of biomaterials. So this has been, of course, a strong, a strong motivation to develop this kind of techniques. And there has been a lot of work on this topic, and around the world, many people have demonstrated that it's possible to image a wide variety of biomaterials using liquid cell microscopy, ranging from full very thick human cells, bacteria, and also smaller entities like viruses and nanometric proteins. And clearly, it's a great technique to image the structure of biomaterials with nanometer resolution. Now, what is also very clear is that the imaging dose, the dose rate that we need to obtain those images, it's clearly above the lethal dose for cells. So we are not yet able to do live imaging of live cell imaging in the liquid cell microscope. So because of this, there is a crucial need to reduce the electron dose and still having a good resolution. So there is a lot of people that work on this problem. But still, as we have such a nice images, there is a lot of question with these images is what kind of dynamical process we can study on biomaterials and how those processes can be related to functional biology. And also, when you work on biomaterials, sometimes you have a lot of problem of visibility when you work on light biomaterials. So there is also a question on how we can improve the visibility of biomaterials in the liquid cell. So to work on this problem, I have collaborated with Florence Gazot and Max Pifou from the MSc lab at the University of Paris. And we worked on the imaging extracellular vesicles in under physiological conditions, so in cellular buffer. And so extracellular vesicles, they are vesicles from micron to nano size, which are involved in the communication between cells. And they have also a great potential for biomedical application ranging from cancerology to regenerative medicine and drug delivery. And at that time, my colleagues, they were developing a high yield and large scale production method to produce extracellular vesicles. And they did a fantastic work, which leads to the creation of this startup, Everzoom, that now commercialize highly concentrated solutions of vesicles that have a good concentration to be able to for therapeutic application. But now, what you see on what we did to image those biomaterials and biosoft materials in the liquid cells, we had, because they have such a very poor contrast, we had to use, we have developed an in situ staining method, which consists simply in adding a little bit of chloric acid in the cell buffer. And you see that you observe the growth of very small nanoparticles at the surface of the vesicles, which slowly increase their contrast. And you can stop the process whenever you run by reducing the dose. And you can, once their contrast is satisfactory, to image their morphology and their dynamics. So it's very useful to image light materials, and I think it can be used on many other low contrast materials in the liquid cell. Now, what we did first, we had to confirm that the object that we observed were not perturbed by the beam or by the confinement inside the liquid cell. So we did a lot of size measurement, and we compare our results with other techniques. And we compare the liquid cell TEM experiment in PBS with cryo-TEM and also nanoparticle tracking analysis, which is an optical method which is widely used to analyze this type of object. And we were very happy to see that the size distribution that we obtained with these three analysis technique were pretty much the same. So we were very happy. We can observe the structure of those materials. But of course, when you do liquid cell microscopy, what is very interesting is to do dynamical imaging. So at first, we have studying the diffusion behaviors of those vesicles. That's what you see on this video. And from this type of video, you can do nanoparticle tracking analysis to obtain the mean square displacement of those vesicles. And it turns out that they have clearly a subdiffusive behavior. You see that the mean square displacement is at least 3 orders of magnitude below what we expect from Brownian motion. So finally, it's a chance, because otherwise it will be very difficult to image those particles. And we wanted to know what affects their diffusion. And here you see the standard deviation of the position of the standard deviation of the position of the vesicles with respect to their mean position. And you see that there is no correlation between the motion of the nanoparticles and their size, but there is a clear correlation between the motion of the vesicles with their signal to noise. So it clearly shows you that what you can see here on this image, you see that the vesicles which have a lot of gold on their surface, they don't move. They are almost motionless in the liquid cells. So from this, we have been able to say that the anomalous subdiffusion behavior of the vesicles is due to the strong interaction of gold nanoparticles with the silicon nitride membrane, which is known to be positively charged when it's irradiated by the electron beam. And then something which is even more interesting for biologists, we wanted to know if we were able to study the dynamics of bilayer membranes. So we try to study the thermodynamics, the thermodynamical shape of those objects as a function of the osmotic pressure. And here I represent three typical morphology of vesicles that you can find in the liquid cell. So you have round vesicles, you have ellipsoidal vesicles, which correspond to a 3D shape, oblat or prolat, and you have also these shape which correspond to stomatocyte if you have only one curved surfaces, or discocyte if you have two curved surfaces. So these are the three typical shapes that you find in the liquid cells. And here you see that if you work at physiological osmotic pressure, you see that the great majority of the vesicles are round, and you have only 10% of them which are slightly elongated. But when you work at high osmotic pressure, you see that the proportion of deformed vesicles become predominant. And this is exactly what we expect from the thermodynamical point of view, because when you increase the osmotic pressure, this type of object, they want to homogenize, to equilibrate the osmotic pressure inside the vesicle and outside, so they release water through their bilayer membrane, which reduce their volumes and lead to their deformation. So we were very happy that we were able to see this type of dynamics in the liquid cells. And in some occasion, we also observe the aggregation of vesicles, and sometimes we also see the formation of fusion pores. So these two examples show you that liquid cell TEM is actually, with liquid cell TEM, it's actually possible to study the bilayer, the dynamics of bilayer membrane, which are extremely important for many cellular process. And I'm sure this is something that might be interested for many biologists. Now, to finish, I wanted to show you some example on the biomineralization process. The mineralization driven by bacteria in biofilms is known to be a crucial process in the cycling of metals in the environment. And so far in the liquid phase TM community, there has been a lot of work on the nucleation and growth of minerals, exactly as in solution, like I show you for the case of gold. You can study different type of minerals formation. But there has been some work also in biomimetic media by adding some polymers that are generated by bacteria in the exopolymer matrix. But so far, nobody has studied the mineralization process directly in the biofilms. And that's what we try to do with collaboration with Alexandre Gelabert and Bénédicte Ménez from the Institut de Physique du Globe de Paris. And so we try to meet this challenge with the idea not to observe live cells. I told you it's not possible, but use them as a template to see the effect of their surface and the exopolymer that they secrete on the for providing nucleation site for mineralization. So we did the experiment on different strain of E. coli bacteria. One, 2K7, which is known to secrete a lot of exopolymers. So you have a bulky polymer layer around this type of bacteria. And we also work on another strain of E. coli, MGF, that is known to secrete no exopolymer, or just not a lot of exopolymer. And what you observe on this image series, you see that the contrast of the bacteria increase over time simply because you observe the formation of a manganese-rich shell around the bacteria. And this has been confirmed by in situ local EDS analysis. So what is interesting in these two image series is that you see that they have a very different growth process on those two strains. On the MGF with no exopolymer, you observe the formation of an homogeneous layer around of manganese at the surface of the bacteria. Whereas on the E. coli with a lot of exopolymer at the surface, you observe the formation of isolated and spherical nanoparticles at the surface of the bacteria, and also in the vicinity of the bacteria in the solution. So we believe that this type of nanoparticles that grow outside not on the bacteria are due to the exopolymer layer that is around the bacteria, because when we do the same experiment now far from a bacteria, we don't see any growth. So it's only in the vicinity of the bacteria that we observe this growth process. Now, this result has been interpreted as an effect of the bacterial surface chemistry, because the bacterial cell surface, it's negatively charged because you have a lot of deprotonated carboxylic group and phosphoryl group at this surface, whereas the exopolymers have only poorly polarized hydroxyl group in the exopolymer, and this negatively charged surface is confirmed when you look at the electrophoretic mobility of these two strains. You see that it's 10 times more negative for MGF, which is the one with no exopolymer, and this negative charge at the surface of the bacteria are obviously cation fixation sites, so nucleation site. And as you have many of them on the MGF strain, you have the formation of an homogeneous layer over this bacteria. While when you work on the 2K7 bacteria, because of this large exopolymeric layer around the bacteria, you have only a few nucleation sites. And that's the reason why you see the formation of isolated nanoparticles. So all those results, they show that there is clearly an effect of the bacterial surface chemistry, and it seems that the charging of the surface play a role on the dynamics of mineral formation. But of course, to have a better, it's a very simple view of what is an exopolymer. So that's why now we have an ongoing work right now on the mineralization, on studying the mineralization process on model systems, and we use those 1 micron large diameter polystyrene beads with various surface functionalization. So we hope that this type of experiment, where we clearly see an effect of the surface functionalization, will allow us to better understand the role of surface chemistry on the growth process of minerals and manganese minerals at the surface of bacteria. And with this, I'm done. So I'm gonna thank all my co-workers, first at the MPQ lab, and all my collaborators in the other labs. And as Kristian told you in its introduction, I want to announce an upcoming conference that will be held at the University of Paris. It's the 5th International Conference on In Situ and Correlative Electron Microscopy that will be organized from September 8 to 10 at the University of Paris. It's co-organized by Kristian Mølhave, Neil de Jonc, and myself. So please visit the website. You'll see that we have a fantastic list of invited speakers, and take the time to submit an abstract in the next 3 weeks. And with this, I'll be happy to answer all your questions.